Hi, I'm Rick Barron, and thanks for joining us again for another segment of RAF5. Today, Dr. Gary Gronseth and I want to talk about the randomized controlled trials of methotrexate and mycophenolate for myasthenia gravis. Interest in mycophenolate began in the late 1990s, and there were several large series published of anecdotal reports which all seemed to indicate that mycophenolate was helping myasthenia gravis patients. Then there was a small randomized controlled trial by Dr. Mariggioli with 14 patients that also gave a hint that mycophenolate might be effective. This was followed by two rather large placebo-controlled randomized trials, one that was investigator-initiated and led by Dr. Sanders and that was federally funded, and the other one that was led by a pharmaceutical company, Aspreva. The Sanders study, which I was involved in designing, was just a 3-month trial, whereas the Aspreva trial was 9 months. To everyone's surprise, at the end of the day, both studies were negative for showing a beneficial effect of mycophenolate in myasthenia gravis. Here is a graph on the top of the Sanders study showing the QMG score. It was essentially the same in both groups. Both groups improved over time, but there was no difference between placebo and mycophenolate. And under that, the similar data from the longer Aspreva study. So why were these trials negative? This continues to be hotly debated in the neurology, neuromuscular world, and I think there are several possibilities. One is that the drug just doesn't work for myasthenia gravis. The other is that all the patients in the Sanders study were on put on the same prednisone dose, 20 milligrams a day, and prednisone may just be so effective in myasthenia gravis that it masked the mycophenolate effect. Maybe the studies weren't long enough, and you can say that about the Sanders 3-month study, but the Aspreva 9-month study you would think was long enough, but some argue it wasn't. Were our endpoints not good enough? Does the QMG, is it not as reliable as we thought, and some of the other endpoints we used, or was there something wrong with the population that was enrolled? Because of the negative mycophenolate study, we then launched a methotrexate in myasthenia gravis study. Dr. Passnor and I in the muscle study group were able to get another federal grant to do this investigator-initiated multi-site trial, and we enrolled 25 patients in a methotrexate group, 25 patients in the placebo group. Patients were all on prednisone, and instead of looking at QMG as our primary endpoint, we decided to look at prednisone dose, using prednisone as a primary endpoint to try to mimic what was done in the old azathioprine study. And the results of this study were recently published in Neurology, and they were presented a couple years ago. And as was similar in the mycophenolate study, this study was also negative. This just came out in Neurology last month, and it showed that if you look at the prednisone, the patients had a forced prednisone taper, but using a standardized protocol, and at the end of the day, there was no difference between the prednisone dose in the patients that were taking methotrexate versus placebo, 13 versus 15 milligrams, which is not statistically significant. And we looked at secondary endpoints such as QMG, MG-ADL, and other scores, and also there was no statistically significant difference between both groups. So once again, you could ask the question, why was the methotrexate MG trial negative? And again, I come up with similar type of answers. Prednisone may work too well, and it's just very hard to do an MG study with patients on prednisone. Again, maybe like mycophenolate, a conclusion is that the drug just doesn't work in myasthenia gravis. Maybe we used too low a dose of methotrexate, but we did use 20 milligrams a day or a week orally. We did have a lot of dropouts in the placebo group. Could that have been a problem? Were we underpowered? And did we do some? Did we just not handle the dropouts well? So I don't have the answer, but I think I'm going to have my colleague, Dr. Gronseth, come up and try to give his insight into why studies like this are so difficult to do and to prove to be positive. Dr. Gronseth.
Thanks, Dr. Barron, for having me back. So some of these issues Rick has already touched on, but there's basically 3 issues that you should ask. And you shouldn't just ask these issues because the study disagrees with what you expected. You should ask these studies regardless. We commonly do that, unfortunately. So the first question is, was there bad study design and execution? In other words, poor internal validity? And the answer for the randomized controlled trials is no. Now, Dr. Marigiroli's study is rated class 2 by AN criteria. Still a pretty good study, although severely underpowered. But the other studies were all class 1. So a very low risk of bias relative to design and execution of the study. The second question, and a common cause for a study to be quote negative, is imprecision, lack of power. And it's straightforward to tell if a study had enough power. You just look at the widths of the confidence intervals. And so, you know, these are supposed to be steroid-sparing agents, so I looked at the difference in the daily prednisone use used, that Don Sanders' article measured that as an outcome, and Mamatha Passnor, that was their primary outcome. And if you look at the difference in daily dose of prednisone, the point estimates is about 2 milligrams less if you were on mycophenolate or on methotrexate, but the confidence intervals are consistent with, in Mamatha's study, with it being almost 7 milligrams less. Is that important? Probably not important, probably not, but that would tell you whether, if this includes something important or, more importantly, excludes an important effect, that the study was adequately precise. What about clinical incomes, looking at the QMG? That was an outcome for all the mycophenolate studies, and if you looked at the difference in QMG from baseline for patients on the steroid-sparing agent versus placebo, those confidence intervals are very narrow, and we're able to exclude a change in QMG favoring the steroid-sparing agents, you know, of less than -2. So that study was sufficiently precise, or all those studies together were sufficiently precise to exclude a benefit. What about methotrexate? Not so much. It's still possible that it was only because of random error or imprecision that we didn't see a benefit, because the study isn't inconsistent with a difference of 5 QMGs, which would be a moderately important effect. So I would argue that we can't really be that confident about that outcome, but certainly you can be more confident about the steroid outcome. And then the last one is poor external validity, which is a synonym for generalizability, or sometimes called directness or indirectness. This is the one where people sort of take advantage and just say, oh, I can reject any study if it's not consistent with what I want, because my patients are different. So these studies show that for these patients, using the drug the way it was used compared to steroids alone in the way that they were used, that the outcomes were not different for that time. They show that. Now you can argue, well, that you know, my patients are different, or there's a subgroup of patients that are different, but you don't know that. Maybe the way steroids are used is different, but certainly the time horizon may be an issue. But the bottom line is, you know, the studies are never definitive, but if you believed that mycophenolate or methotrexate worked once these studies were published, your confidence in that belief should be lowered. Now, it doesn't mean that it doesn't work, but your confidence should be lowered. Thank you.
So that's the conclusion. Neurologists should come out from that. The confidence is lower. Still a possibility that they work, but we couldn't prove them using traditional study design.
Right. It's less likely that the drugs are effective. Now, if your confidence hasn't changed at all, or looking at all the articles that were published after,
because we have some of our colleagues that their confidence has not changed at all.
Right.
That increased.
Exactly. And I was going to make that point based on all the articles, and we call that cynicism.
Oh.
Okay. Then there was no point in doing the studies in the first place.
All right. Well, thank you, Dr. Gronseth, once again for being my friend on REF5.